Good morning, sir. Vakira, sir. Good morning, sir.
Good morning, sir. Vakira, sir.
Good morning. Hello.
Good morning, sir. We will start.
Just I think getting connected. It's just maybe take another 2 minutes. I think.
Okay, sir. We will sharply 9:30. We will start it. Thank you. Thank you.
ಇವಾಗ ಸರಿ ಆಯ್ತು. ಟಾಯ್ಲೆಟ್ಸ್ ಅದೇ ಕ್ಲೋಸ್ ಮಾಡಿಬಿಟ್ಟು.
Now you can ask.
I am ready. Participants only 33.
33, sir. Good morning, sir. Vakira, sir.
Oh, good morning. Very good morning, Dr. Niyas. Yeah.
Sir, participants join one by one, sir. We can start, sir. Let them join, sir. According.
You want to wait for some more time till more participants join, or what?
They join one by one, sir. Let we start at 9:30. Let them to join, sir.
Yeah, yeah. I can wait for a few minutes. Fine.
Okay. Sure. Thank you, sir.
They are telling we can start. They are telling we can start. But you can start. No problem. I am ready.
Okay, fine, sir. We can start, sir. Because some people are waiting in the YouTube live streaming. That's why we can start now.
Oh, okay. No problem.
And if we have sufficient numbers, sir, please.
Okay. No problem.
Mr. Sivalango, you can go ahead, Professor. Sivalango.
Okay, can I start?
Yes. Yeah, very good morning and warm greetings to all of us on behalf of PG and Research Department of Biochemistry, Sacred Heart College, Autonomous, Tirupattur. I take this opportunity to welcome you all to Technical Session 1 of Day 2 Science Academies Virtual Lecture Workshop on Recent Trends in Biological Sciences. I have a great pleasure to introduce our resource person of this session, Dr. D.J. Bakiraj, NASA Honors Scientist and Chairman. Center for Natural Biological Resources and Community Development, CNBRCD, Bangalore. Even though we introduced our professor in yesterday's session, it is my privilege to say something about his expertise.
Again.
Sir has been a fellow member in Indian National Science Academy, National Academy of Sciences, National Academy of Agricultural Sciences, the National Academy of Biological Sciences, the Society of Agricultural Biotechnologists, Indian Phytopathologist Society, and the Association of Microbiologists of India. Sir has won prestigious national awards like SR Vyas Memorial Award, So Memorial Award, Dr. G. Rangaswamy Memorial Award, Professor T.S. Sadasivam Lecture Award, Dr. R.Y. Roy Memorial Lecture Award, Dr. Natarajan Memorial Lecture Award, recognition award by NABS, recipient of Distinguished Asian Microbiologist Award 2019 from Asian Microbiological Association and several Lifetime Achievements Award. Sir has served as an advisory committee member in ICAR, DST, DBT, CSIR, ICFRE, UGC, NBA Rubber Board, Spices Board, IFS Sweden, AECR Australia. Dear participants, we have a great eminent personality with us for this lecture workshop, and with his brief introduction, I take this opportunity to thank him for accepting our invitation to deliver a lecture on ecology of arbuscular mycorrhiza fungi. I request the participants to mute their mic when the lecture is going on. Kindly type your questions in the chat box. You can also raise your questions after the end of the lecture to the resource person. I thank you all for patents hearing and have a fruitful day. Now hand over the session to you, sir. Thank you.
Introduction. And shall I start?
Yes, sir.
Am I audible?
Yes, sir.
And this, you can see the screen also?
Yes, sir.
Okay. Okay, the topic I am going to talk today is on ecology of arbuscular mycorrhizal fungi. Yesterday, I talked about the importance of arbuscular mycorrhizal fungi in sustainable agriculture. So this will be on the ecology of it. And yeah, you know that nearly 90% of the vascular plants have the arbuscular mycorrhizal association. Nearly 90% of the vascular plants. And approximately, there are about 300,000 host plants. And there are only 230 AMP species. Just a little mathematics, you divide 300,000 by 230. That means each mycorrhizal fungus can form symbiotic association with 1,304 potential partners. Okay, so that is how people have been describing them as not host specific, though I told yesterday why we have introduced a new term, host preference. And they're all obligate symbionts. Okay, they need the roots of a living host to grow and multiply. And they are ubiquitous. This is a very important point. They are present everywhere, all over the world, in all the continents, in the desert environment, in the aquatic environment. So they are present everywhere. And what is the reason that they are present everywhere? The ecologists were trying to find and give a reason why they are present all over the world. In every soil they are found. Finally, they came with a particular theory. This fungus must have existed in Gondwana land. What is Gondwana land? Before the land got dissected or separated into different continents, as we have now, Australia, America, Asia, and so on. So before this sort of the intercontinental drift took place, this fungus must have existed in one block of land. And So this is what is Gondwana land. All the land together put in together. And when the continental drift took place, then we have the various continents separated as Africa, South America, Asia, and so Australia, and so on. So the fungus got into the various continents. So this is the theory put forth by ecologists that this fungus existed in Gondwana land. Okay, yesterday I told about the produces vesicles, it produces arbuscules, and to which fungal phylum it belongs to, and so on. And I also told how do they improve plant growth. Just to recollect, they improve the uptake of nutrients, especially nutrients which are immobile in soil, and also synergistic interaction with beneficial microbes, protect the plants against diseases, and also protect the plants against water stress, and they produce growth promoting substances. So this is how they improve plant growth. With this background, what I told, just to recollect, let us go into the ecological part of it. Okay, all the workers, yesterday also somebody asked this question, I briefly answered this. People, the giants in mycorrhizal research in till 1990s were telling mycorrhizal fungi never colonize aquatic plants. If you see all the reviews, all the research papers, that is a statement made. And there is one lake very close to the university in Bangalore where I am working. It is called Hebbal Lake. So, which has so many aquatic plants. I told the students, go and collect the plants from this aquatic lake, and then bring the plants. Let us see whether it is mycorrhizal. They brought 27 different aquatic plants, and we had a very nice botanist in the botany department, and she could identify all the 27 species. And we stained the roots for mycorrhizal association. And we were surprised to see that in 3 of them, Cynotus cristata, Salvinia cuculata, and Eichhornia crassipes, that is the water hyacinth, which grows so luxuriantly in all lakes. And so all these 3 were colonized by mycorrhizal fungi. So I told the students, I will come with you next time, and then I would like to collect these 3 plant species myself. And not that I doubted them, but anyway, just to make it sure. So I went to the lake along with the students and collected these 3 plant species. We brought them to the lab and stained. They were beautifully colonized by mycorrhizal fungi. We didn't stop there. I asked the students to collect these 3 plant species from lakes in and around Bangalore. And they brought the samples and they were all beautifully colonized. So we wanted to send this for publication. It went to Transactions of the British Mycological Society, which was published from UK. And somehow they were not very happy with the publication because it's contradicting what the giants in mycorrhizal research were telling. It went for referring to Dr. Barbara Mose, and she called me and she said, beautiful work. Then the next question, are you sure they are from aquatic environment? I said, Dr. Barbara, I am sure, I have collected it myself. So it was going forth and coming back. Finally, it was published. And now, which became the world first report that aquatic plants can be mycorrhizal. Okay, these 3 plant species are free-floating aquatic plants. What I mean by free-floating, the root system is in water, but the shoot portion is above the water level. And you can see that they are beautifully producing flowers and so on. They are the free-floating aquatic plants. It so happened that I had a visiting scientist fellowship to work in New Zealand for 3 years. So I went to the lab in New Zealand. The microbiology department was next to the aquatic botany department, it so happens. And there was one Dr. John Clayton who was in charge of this aquatic botany department. And outside the department, aquatic botany department, he had small tubs containing the various aquatic plants occurring in New Zealand. So when any visitor comes, he used to show these are the aquatic plants occurring in New Zealand. So I went and told John, can I take some samples and look whether they are mycorrhizal? Then he said, no, we have all done that and we have checked whether they are mycorrhizal. They are not colonized by mycorrhizal fungi, I am very sure. Then I was a bit hesitant to again press him. And then one day, the coffee time, the morning, I told him, why don't you give me a chance? Why not I take and then look for and confirm myself? Then he said, okay, go ahead and then collect the samples. So I collected all the plant samples at the roots and looked for mycorrhiza. To my surprise, 2 of them were beautifully colonized by mycorrhizal fungi. One was Myriophyllum triphyllum, other one was Ranunculus rivularis. The difference here is these plants are submerged aquatic plants. These plants never come above the water level. They are 2 to 3 meters below the water level, the entire plant. Okay, so they are the submerged aquatic plants. So when I told this, and he also saw the slides, and he said, you must be having green fingers for mycorrhiza. When I have tried, they were non-mycorrhizal. So he was so much excited, and he took me in the boat with the scuba, that is a diving dress. And I said, I can't dive. So we went to the ocean, and he used to dive and then bring the samples. And I used to take the roots and put it in this glass tubes and bring it to the lab. And we checked it. Even in ocean water, they were mycorrhizal. And again, this was published in the International Journal of Aquatic Botany, which again became the first report that submerged aquatic plants can also be colonized by mycorrhizal fungi. But the New Zealanders went one step further. What they did, they gave this as a thesis problem to a student. They wanted him to, in aquarium, we grow certain aquatic plants. So they asked him to inoculate the aquatic plants in aquarium with the mycorrhizal fungus and see whether it improved growth. And it did increase the growth of the aquatic plants. That was the boys' student's thesis, for which I was the external examiner also. So now we know that aquatic plants are also colonized by mycorrhizal fungi. Another interesting observation or the reports made from the Western world that the mycorrhizal fungi sporulate in summer. So that is all the reports were telling, that they sporulate in summer. So out of curiosity, I told my students, let us collect the soil samples every month and look for the spore numbers. And to our surprise, it sporulated maximum spores we got in winter, just opposite to what was reported. So both of us are correct. The temperate region, the temperature in summer is something more or less equal to the temperature what we get in winter. So that is how the temperature is very important. It sporulates in tropical countries in winter and not in summer. And we just collected the samples, and we also looked for the AM population. How long can it survive? When once we have the mycorrhizal inoculum, how long can it survive? So we took the samples and then placed one set at refrigerator, 4 degrees Celsius, one at around room temperature, 25 degrees Celsius, other one 40 degrees Celsius. So we maintained them at 3 different temperatures. And every month we were looking for the infective propagule numbers. And to our surprise, even when it was kept at 40 degrees Celsius, they were viable for 24 months, 2 years. So what does it message we could get from this? There is no need for storing mycorrhizal fungi in cold room or refrigerator. You can very well keep it in room temperature and they will be viable for 2 years. And another thing which we were wondering is, These fungi are ubiquitous. They are found everywhere. That means they must be having very active methods of dispersal. So either the dissemination of these fungi must be active or passive or both. So we wanted to check this, whether they are actively disseminated. So next to the university, there was an Amco battery company. And so I just went to the company people and then asked, can I take few Amco battery boxes? They said, take as many or as much you want. We wanted to get rid of these boxes. So they were very happy when I brought these boxes. And what we did, this is one battery box, which I am showing here. We separated them into three portions. And you can see we put a wire mesh and then separated this portion. And after the wire mesh, we introduced a nylon mesh. Okay, so that's how we have three different compartments in a single Amco battery box. And the size of the nylon mesh, the pore size was such, it will not allow the roots to pass through. It will allow only the hyphae of mycorrhizal fungi to pass through. So this was the way which we planned the experiment. So in the compartment A, we planted a mycorrhizal plant. And the middle compartment B, we just filled with soil and no plants. And in compartment C, we planted an uninoculated non-mycorrhizal plant. And we were waiting. And of course, the whole thing was covered with sterile cotton. And so that, and we also took care to water it with sterile water. And then so that what we were waiting is how long it takes for the non-mycorrhizal plant to become mycorrhizal. How can it become mycorrhizal? The hyphae from this side has to travel all the way through compartment B and then come to compartment C, and then only it gets colonized. So we were eagerly waiting and taking samples periodically, and this is the result. So how much time it took? It took 180 days for the non-mycorrhizal plant to become mycorrhizal. And what is the distance between these two plants? It was 300 millimeters, that is 30 centimeters. So to travel 30 centimeters is 1 foot. Okay, so it takes 180 days to travel 1 foot. So that means it can travel only 1.66 millimeters per day, spending its own ATP. So that is not the mechanism by which these fungi can disperse, and they are present everywhere in the world. So active dispersal is not the method by which they spread actively. So then what is the next choice? It's only passive dissemination. So we looked into the passive dissemination. There are plenty of earthworms in the university campus. So earthworm cast. So we just took the earthworm cast and looked whether it has mycorrhizal propagules. And we also took them at different intervals, at different monthly period. And you can see that maximum number of infective propagules were present in the month of October. And then the next question is, how long can it survive? It had plenty of mycorrhizal propagules in the earthworm cast. How long can it survive in the earthworm cast? So the viability was determined after air drying the earthworm cast, and every month we used to take the sample and look for the infective propagule numbers. And we could see that they remained viable for 11 months. Okay. So, but from literature and by discussion with our zoology group, we could see that earthworms bring 2 to 5 centimeters of soil to the surface, very small amount in a decade. Okay, very small amount of soil. But when once it comes above ground, it can disperse horizontally through water, through air, and so on. So makes it easy for them to spread. So that the earthworms earthworms are one of the important disseminators of fungi. Next, lot of millipedes are also present in the campus. So this was the particular species, Philanogrostreptus nigrolabiatus. So that was the species predominant in our campus. So what you are seeing here are the fecal matter of the millipedes. So we took the fecal matter. In fact, I should tell here, the fecal matter looks so attractive. It looks like small sweet which we give to children. It looks so good. And so we brought this fecal matter and then examined it for the mycorrhizal population or infective propagule numbers. Okay, what is the result? They were present. The propagules were present in the fresh fecal pellets, but they lost their viability in 4 days of storage. So we are breaking our head how to interpret this. Again, discussing with a zoology group, we could, and finally going through literature, we could see the digestive tract of these millipede. The pH was 4 and it has several digestive enzymes. So probably this is the reason why they lose their viability in 4 days. And next is termites. Plenty of termites in the campus. So you know that termite mound, it is dry in some places, the tip is wet. So the wet portion and the dry portion. So we brought the samples from the termite mounds and then looked for the presence of mycorrhizal fungi here. So this is the, and again, there are 2 types of termites which build their mound above ground. There are some termites which remain only below the soil, which we call them as subterranean termites. We took the samples from the above ground mound building termites as well as the subterranean termites. In the mound building termites, we took it from the dry termitaria as well as the live termitaria. And these are the number of samples which we have examined, and look at the spore numbers, plenty of spores, and the infective propagule numbers. Okay, so look at the difference here. The spore numbers were very high, 160. When we look at the infective propagule numbers, it was 0. What's the difference between spore numbers and infective propagule numbers? Infective propagule numbers give only the viable count of the mycorrhizal fungi. Whereas the spore numbers, the spores may be dead or alive, but it gives the total spore numbers. So the spores are there plenty, they were all dead. So the infective propagule numbers was 0. Whereas the soil which was away from the termite mound, it had the infective propagule of 200, which very, very clearly showed in termites, there is something that is killing them. Okay. So again, going through literature and having discussion with the group of entomologists and zoologists, we could find that this saliva of termites contain a compound called benzoquinol, and benzoquinol is antifungal, and that is how it is killing the am fungi. So termites are not good disseminators of am fungi. Then next step, looking at ant hills. So plenty of ants everywhere. So the ant hill samples we took and then looked for the presence of mycorrhizal fungi. And this is the result. And we took the samples from different locations, from the root zone of Lucina, Goa, grass, and so on. And look at the infective propagule numbers, so high. And again, going through literature, we could see ants bring annually very little quantity of soil above ground, only 0.11 kg soil per meter square to the surface. But when once it comes to the above ground, it comes to the surface, there are plenty of chances that it can be further transmitted through water, through air, and so on. So ants act as very good disseminators of am fungi. So what happened was, we had a project funded by DST on ecology of am fungi, and there was a research scholar working on this particular project. So he was discussing with me as to what will be the next experiment that we have to do. So when we were discussing, we could see one wasp coming and building the nest in my office room. So this boy asked me, "Sir, will mycorrhiza be present in the wasp nest?" I told him, "I really do not know. We have to look whether it has mycorrhizal fungi." So that became very interesting. He took the sample, the wasp nest, and looked for the presence of mycorrhizal propagules. And it had plenty of mycorrhizal propagules. Okay, so that gave us an idea to collect all the wasp nests. And again, our entomology people helped us. And these are the various wasps which we studied: Styriform Madras Patnam, and Styriform spinole, Eumenes conica, and Rhynchium species. These are the 4 different wasp nests which we looked into. And see, the plenty of infective propagules were present here. Of course, in one we couldn't see any. And the Rhynchium species, of course, the number of samples we tested was also less compared to the other ones. So from this, we could conclude that wasps play a very important role as disseminators of fungi. Again, going through literature and again with the discussion with the entomology group, we could make out that a female wasp can carry 40 milligrams of clay, small quantity, but it can travel 335 kilometers carrying this clay. That means they act as excellent disseminators of fungi. Okay, this is about the dissemination of fungi. Here I would like to add one more thing. And we had one Indo-US collaborative projects with the, and at Corvallis and Oregon State University. And this project was for 5 years. And the agreement was that every year I go and spend 3 months in Oregon State, and the scientists from Oregon State come and then spends another 3 months in India. So one of the visits when I went to Oregon State, that was a person who was very much interested in forest plants. So he took me to the forest in Oregon State. I was surprised to see a board there, a notification to the public saying that rodents are excellent disseminators of mycorrhizal fungi. Do not kill them. See to what extent Americans have gone. They have understood the importance of mycorrhizal fungi. So another thing which we experiment, yesterday I think one student or one teacher asked me a question. Is it necessary to inoculate mycorrhizal fungi every season, or when once we inoculate, will it persist for a longer period? This is the question yesterday one of the participants asked. And we had an experiment to check this. Okay, so here we had certain uninoculated pots, and we did this in the as a pot experiment. Later it was taken as a field experiment. So we had one treatment was uninoculated, and the second treatment inoculated in the, we know there are 3 seasons in a year. So inoculated only in the first season. And another treatment inoculated in the first season and the second season. And the third treatment inoculated in the first, second, and third season, all the 3 seasons. And the fungus which we used for inoculation was Glomus intraradicis. We particularly chose this particular fungus because that was not present in the university campus soils. That is the reason why we chose this, and see how it works. And this was the result. What is the result? It's inoculated in one season, infective propagule was so less, but infected in all the 3 seasons, you can see the infective propagule number was very high. So this shows for a fungus which is not native, which is not existing in the soil, it is necessary that we inoculate all the seasons. But the story may be different if we work with a fungus which is already present in the soil. And that we have not done, and it's worth doing that. And this mycorrhizal fungi, the spores of mycorrhizal fungi has an enzyme called malate dehydrogenase. And so we isolated this particular enzyme, and this enzyme has got 3 isozymes, okay? 3 isozymes. And we did the polyacrylamide gel electrophoresis for the presence of these 3 isozymes. And this is the standard malate dehydrogenase. I can see it showed after the electrophoresis 3 distinct bands. And this is the sample which was inoculated for all the 3 seasons. You can see the bands only here when they were inoculated for all the 3 seasons, confirming what we have said earlier. Whereas where the plants were inoculated only for 1 season or 2 seasons, the bands did not appear. Okay, another interesting collaboration we had with the genetics and plant breeding department people. And in the campus, we have the genetic resource of various crop plants. So we took cowpea as one of the resource. So they had, the genetics and plant breeding department had 101 genotypes of cowpea in the garden. So I went and talked to them. Can I take the root sample of all your samples and look for mycorrhizal colonization? And they were very happy. Go ahead and do it. So we took for the colonization, and we could see they were all colonized. And though there was variation in the percentage of colonization. And finally, the genetics people came with certain conclusion. They looked for, analyzed for the genetic coefficient of variance. There is a certain formula for doing this. Genetic coefficient of variance was very high, which indicates a character which can be used in selection and breeding of crop improvement programs. And they also looked for the heritability. The heritability was also very high. What does it mean? Higher the heritability of a character, it is better for plant breeding. And they also looked for genetic advance. Genetic advance was also very high. What does it mean? It suggests how much better the progeny is compared to the parents. So from this, we could conclude that fungal colonization, it is a heritable trait. It can be inherited from the parent to the offspring. So the plant breeders, when they are breeding, when they can look, one of the characters they can look for, plants with high mycorrhizal colonization. Okay, this is a very interesting result we could get from this particular experiment. Another interaction study which we did was very few studies on yeasts, soil yeasts. So we thought it will be interesting to see whether soil yeasts have some effect on mycorrhizal association. So we tried various soil yeasts, and we could see that Rhodotorula mucilaginosa, when inoculated with Glomus mosaic, increased significantly the biomass yield of the plant. This was again done with cowpea, and the nutrient content, and increase the mycorrhizal colonization. And the yeast population was also high compared to other treatments. So that means soil yeast has a synergistic interaction with mycorrhizal fungi. Very few studies, I can say only probably this is the only study, or I see one more study. Apart from that, there's young people have a good opportunity to study this particular aspect. Another student wanted to do her project work in my lab. She came from Mangalore University, and she was interested in vermi wash. You know what is vermi wash? Vermi wash is when earthworms are allowed in lukewarm water, the hot water light which they can tolerate, they just exude their body content, which is referred to as vermi wash. We again put them in cold water and they become normal. So what she did was she was soaking the seeds in vermi wash and then sowing the seeds. And what we found was the number of nodules and nitrogen fixation increased when the seeds were soaked in vermi wash, which we never expected. And still today, we do not know what is the mechanism by which vermi wash increases nodulation and nitrogen fixation. One step further we went, when the seeds were treated with vermi wash and also given the plants given vermi compost in the substrate, the mycorrhizal colonization tremendously increased. Okay. Again, we do not know the mechanism by which vermi wash is increasing not only nitrogen fixation, but also colonization by mycorrhizal fungi. And coming to some generalizations, what is most common genus present all over the world, including the Indian soils? I told yesterday, Glomus. Glomus is the widely distributed fungus. And in tropical soils, Glomus and Gygospora are the most common. In soils which are acidic in pH, around 5 and little less than 5, Acylospora laevis is the dominant fungus. In soils with higher pH, Glomus mossae dominates. And next, the various agricultural practices have an influence on mycorrhizal fungi. Again, being in an agricultural university, we were very much interested in this topic as to how the various agricultural practices influence mycorrhizal fungi. So we have looked into the various aspects like cropping pattern, crop rotation, tillage operation, organic amendment application, fertilizer application, and crop protection, and so on. So I will just go into each one of them briefly. Here we had a monocrop of maize, and we also had a monocrop of soybean. And so we looked into the infective propagule numbers. And then we also had an intercrop of maize and soybean. So you can see here, the infective propagule numbers were much higher in the intercrop when maize and soybean were grown together compared to only the monocrop of maize or soybean. So what is the reason why in intercrop it is high? Probably in intercrop, the root density is much higher. There's more chance for the mycorrhizal fungi to colonize the root system because two different crop root system come in there. And moreover, there two crops take away more nutrients from the soil, and mycorrhizal fungi work very well in soils which are less in nutrients. So these could be the reasons why it is high in intercrop. Another is crop rotation. We know the farmers take one crop in the first season, second crop, and 3 seasons, they have 3 different crops. So we had this experiment on crop rotation. Finger millet, that is ragi, was grown in the first season in all the treatments. And in the third season, cowpea was grown in all the 3 seasons. Both finger millet and cowpea are mycorrhizal hosts. Okay, they always have mycorrhizal association. But there are some plants which do not form mycorrhizal association. Okay, and plants belonging to Brassicaceae, as I told yesterday. So what we did was, the first treatment, we had the mycorrhizal host in all the 3 seasons, finger millet, cowpea. And in the second treatment, finger millet, mustard, which is a non-mycorrhizal host, then cowpea. Third treatment, finger millet, no crop, left fallow. Okay, sometimes the farmers leave their land fallow. And the third season, we took cowpea. And what is the result? Look into the infective propagule numbers. When the mycorrhizal plant is grown in all the 3 seasons, infective propagules was very high. When a non-mycorrhizal plant was grown in the second season, the propagule numbers came down. And when the land was left fallow, still the propagules came down. So this was the conclusion we could draw. Another experiment was done was the influence of alley cropping on AMP fungi. What is alley cropping? This is a very common practice followed by farmers in Africa. Between food crops, they grow a tree crop. After a few rows of food crop, they grow a tree crop. So this is becoming common in India also nowadays. So what we did was, in this experiment which was done, and we took 2 tree species, Lucina leucocephala and Gliricidia sepium. Both are green manure plants where we can cut the leaves and apply to the soil. And these tree crops were grown at the spacing at 4, 8, and 12 meters between the 2 trees. This was the spacing. And what are the field crops we took in between the trees? Finger millet, ragi, that is peanut, groundnut, and pigeon pea, and the red gram. And And we took the samples and looked for the infective propagules. So of the two, we can see the result here. Of the 3 crops species which harbored the maximum infective propagules, it was finger millet at the maximum number of infective propagules compared to red gram or groundnut. Of the 2 tree species, Lucina harbored maximum number of infective propagules compared to Gliricidia. About the spacing, when the tree trees were placed 12 meters away between the trees, we could get maximum number of infective propagules in the croplands. So this is the conclusion, which gives us some idea how we can plan our agronomic practice. Another project we had, which was funded by United Nations Environmental Program, and they were interested in seeing the mycorrhizal population in coffee ecosystem. That was done. This was done in Kodakurg district of Karnataka, where a lot of coffee is grown. And we have, when farmers grow coffee, and they grow certain shade trees between coffee. And there are 2 types of shade trees, evergreen trees and deciduous tree species. And again, some farmers grow only 1 type of shade tree. Some farmers grow 2 or 3 types of shade tree. Some farmers grow varieties of shade trees in coffee plantation. Again, there are 2 types of coffee, Arabica coffee and Robusta coffee. And we were taking the samples and looking for the infective propagule numbers. What are the conclusion? Coffee under evergreen ecosystem had higher infective propagule number. Of the 2 coffee types, Arabica supported higher infective propagules compared to Robusta. Of the 3 shade typologies, coffee grown under 2 shade tree species had the highest number of infective propagules. So this is a system which was very helpful to decide which is how many number of shade trees should be grown, which is the better coffee type to be grown, and so on. Next thing we looked for was the tillage. We know earlier farmers used to use the drawn plows and then plow the soil. Now tractors have come. So we compared these two, and tillage, deep tillage, as we do in case of tractors, compared to the bullock drawn, and see which is better. Naturally, the bullock drawn less tillage is better for ant colonization, because the deep tillage disturbs the hyphal network and therefore it disturbs the mycorrhiza. And this is from the literature I could take this. And again, comparing the different tillage types, the conventional tillage, this is the old type of tillage in rice crop, it had a positive effect of ham fungi. Deep tillage had a negative effect. In wheat crop, in between the rows, some farmers do the mulching, put the straw or polythene sheet, which had a positive effect on mycorrhizal fungi. Some farmers burn the straw. That had a negative effect on ham fungi. And coming to the organic amendments, people do add lots of organic amendments to the soil. We tried 3 different types of organic amendments: paddy straw, maize straw, Pangamia leaf. And of these 3 amendments, you can see Pangamia leaf gave the maximum number of infective propagules, probably because it is a legume compared to the other 2, which are cereals. And green manuring is very commonly done by farmers. And we tried 3 different legumes, Dolichos, Cannavelia, and Crotalaria, and this was done in coffee plantation. And we looked for the infective propagule numbers. Again, of the 3 legumes, Crotalaria supported maximum number of infective propagules. And again, many people say that mycorrhizal fungi are reduced by the application of fertilizers, but they are increased by the application of compost or farmyard manure. It is true. In this particular experiment, we could see this. The control had only 0.065 infective propagules. But here, when the fertilizer was added, you can see it was brought down a little bit. But when the farmyard manure was added, it was increased. And we have gone a little further to see the bad effects of chemical fertilizers. You know, the fertilizers, especially in the tropical soils, our soils are deficient in phosphorus. When we add P fertilizer, and especially heavy doses, it decreased fungal population. But we have some so slowly released phosphatic fertilizer like rock phosphate, bone meal. They enhance mycorrhizal colonization. Regarding nitrogen fertilizers, heavy doses had a negative effect. And again, nitrogen is applied in the form of nitrate or ammonium form. Nitrate form was more inhibitory compared to ammonical form. And regarding potassium, I could get only one reference from Colombia. They have added up to 200 kg per hectare, and that increased the AMF colonization. And here again, the effect of fertilizers on fungi, which we could do in very small tubes. And here you can see the percentage colonization in when no fertilizer was added was 95% colonization. When recommended level of phosphorus was added, it came down to 74. When double the amount was added, you can see it has a very deleterious effect, but 50% had not much effect compared to control. So heavy doses of fertilizers have a deleterious effect. And we also, in the university campus, we had an experiment on long-term fertilizer application. That means the fertilizer is applied to the same plot year after year, year after year. The crops taken may be different, but the year after year, the same fertilizer is applied to the same plot. And this was taken after 16 years. Okay, here with recommended level of NPK fertilizer, the effective probable number was 540. When higher dose was added, it came to 187. When 50% of NPK was added, it was very high. So that is why adding 50% of the recommended dose has no adverse effect on mycorrhizal fungi. Another experiment we could do was on soil solarization, because farmers sterilize soil by solarization before taking up the nursery. So here we used two thick polyethylene sheets of two different thickness, 0.1 mm and 0.05 mm. These were the two thick, the polythene sheets were of two thicknesses. And solarization we could do for 20 days, 40 days, and 60 days. What was the result? And when we used 0.10 mm, okay, and when that was more effective, which killed the fungi. And of the 3 days, we could see that solarization for 60 days was more effective. It almost removed the mycorrhizal fungi. So it was completely sterile. And coming to pesticides, yesterday again, I think somebody was asked about the pesticides. And here I was told that some pesticides have no effect. Most of the pesticides inhibit, and some of them stimulate. I'll just give some examples. Coming to fungicides like pentachloronitrobenzene, binomil are inhibitory, whereas captan and rhinan have no effect. Coming to insecticide, metacistox and aldrin are inhibitory, whereas chlorpyrifos and sumicidine have no effect. And DBCP stimulates mycorrhizal colonization. Coming to fumigants, all the fumigants studied so far are all inhibitory to fungi. Coming to herbicides or the weedicides, glyphosate, ethyl are inhibitory. 2,4-D, which is very commonly used, of course now banned, and carbendazim have no effect. And anilofos stimulates mycorrhizal fungi. So this is in a very brief way, I have just put this data. Coming to lime application in acidic soils, to bring the soil pH up, farmers do the lime application. So what is the effect of lime application on fungal population? It has been studied by 3 different groups of mycorrhizal scientists in 3 different countries. The results are different. One reported that it reduced mycorrhizal fungi. One reported that, another group reported it has no effect. The third group reported it increased mycorrhizal fungi. I think we need more studies to draw the definite conclusion. And here again, we had another project funded by the United Nations Environmental Program, and where we were asked to study how the land use types affect mycorrhizal fungi. Okay, this work we did in Western Ghats against various land use types. We have the natural forest, we have the grasslands, acacia plantations, cardamom plantations, coffee plantations, and paddy fields. So we looked into the mycorrhizal propagule numbers, and we could also isolate different mycorrhizal fungi from these and different land use types. We could isolate 282 isolates of fungi from these and try to identify most of it. And fungal species and diversity, species diversity and richness was also studied. The species diversity index, that is the Shannon-Wiener index, which is used for comparing the diversity between various habitats, was also studied. And we also studied the Jacquemin's index, that is the species richness index, which gives the number of species in a community. The results are given in the next table. So when we see Shannon-Wiener index as well as the Jacquemin's index, both were very high in natural forests. That means not disturbed by mankind. And same thing was true in case of grasslands also. It was very high where it was not disturbed by human beings. When it was when it was to some extent disturbed by acacia plantations, cardamom plantations, coffee plantations, the indices came down. But in paddy fields, it was the least, because there the interference by the humankind is maximum. Okay, and thereby, which very clearly showed that in the different land types, when they are not disturbed, the indices are very high, the diversity of fungi is high, but when there is disturbance by human people, then the diversity comes down. And this is one method of agriculture which is done in the northeastern part of our country, that is the northeastern states, which they call as slash and burn agriculture, or they also call as zoom cultivation. What they do, they burn the forest and then raise the crop for a few years, and then they leave that and then go to a new forest area, burn it, and then take the new crop. And this is the information we could get from the Arunachal Pradesh group. And here, the total number of species which they could isolate from the natural forest as well as the zoom cultivated area, 6 different genera of fungi they could isolate. But what happened when they looked into the species level? 10 species were totally eliminated because of this slash and burn cultivation. 10 species of fungi were completely lost. That is mainly because of the burning of the plants. And that is the main reason. And this is a work which I could see done in Japan. Very interesting work. That's why I just included this. They took, this work was done on a hillock, and one side of the hill was ending in the ocean, seaside. That's what they called as seaward slope. Other side of the hill was ending in the land, so which they called as landward slope. In the seaward slope, the grass that was predominantly present in the hill was Elymis and Elymis mollis. And in the landward slope, it was Miscanthus sinensis. Okay, they collected the roots, extracted the DNA, PCR, followed by PCR sequencing, and then did the community analysis. What they could say is, they could isolate 22 fungal species belonging to 5 families. So they could see that the diversity of fungi was much higher in the grass growing in the seaward slope, that is Elymis mollis. But these fungi were less in their grass growing on the landward slope. So the reason the authors have given is probably it is because of host preference. Probably the fungi prefer Elymis mollis as a better host compared to the other one. And another reason they have also said because of soil disturbance, because frequently the waves of the sea come and disturb the soil. So maybe soil disturbance could be another reason. So this is a very interesting result. And perhaps before conclusion, I just want to introduce this particular slide. And we have one Indo-Swiss collaborative project which is ongoing. And part of the work is done in Switzerland and part of the work we are doing here in India. And it is done in two different places in India. And here, yesterday again, one of the participants asked a very interesting question. To answer whether two plants are connected by mycorrhizal fungi. Will it happen? Yes. And I told, yes, it can be connected. So this is the experiment that is going on. I will tell you. The two plants which we are doing in this particular project is which is done in dry land agriculture where the moisture is very less. And we have taken two crops. One is a deep-rooted crop, that is red gram, pigeon pea, which the tap root system is it can go to the very deep layer. And other one is ragi, the finger millet, which is a shallow-rooted crop. Okay. And what we have seen here, and this is a dry land soil, and the deeper layers are wet. Okay, there is moisture in the deeper layers, and the yellow portion is dry. The soil is very dry. We have inoculated the pigeon pea with the mycorrhizal fungus. Okay, we inoculated with the pigeon pea with the mycorrhizal fungus. So this is colonized by the mycorrhizal fungus, and the fungus, the common mycorrhizal network can spread, okay, as we have shown, and it can colonize finger millet. And it waters, the water from the deeper layer is irrigated through the hyphae to the shallow-rooted crop. This process is now called as bioirrigation. Okay, this was shown through laboratory microcosm experiments in Switzerland to begin with. Okay, how they have done? They took containers, two containers. One is a large container in which they have added gravel and so on. Finally, some sand here at the top. And there is another container below, and this is porous. Okay. And here, and in the bigger container, they introduced a smaller container. Smaller container is a bag with perforations in it. Okay, with holes. The size of the pore here will not allow the roots to go in, but only the hyphae of mycorrhizal fungi to go in. So what they have done, they have inoculated this is pigeon pea, which is a deep-rooted plant, pigeon pea, and they have inoculated with the mycorrhizal fungus. Okay, that means this is mycorrhizal, and the hyphae from the pigeon pea whether it can enter in through the holes and then colonize finger millet. That is what they have seen. Yes, it has colonized. In addition to that, one more positive confirmation is that here they have treated with deuterium labeled water and then looked for the hydrogen isotope in the root system of ragi. And they could trace the isotope in the root system, which very, very clearly showed the hyphae from the deep-rooted plant can transport water to the shallow-rooted plant. So based on this, the first experiment that was done in Switzerland, now we are doing these experiments first under polyhouse conditions. Now it has gone to the field experiment. So this is the type of work which we are doing at present. So mycorrhizal fungi can help in irrigating shallow-rooted plants under dry land agriculture conditions. Okay, so now we will come to the conclusion. Now it is very well established that AMP fungi improve plant growth. But what is one of the impediments coming in the way of field application is the very little information we have on ecology of these fungi. Again, I would like to emphasize and say, lot of studies on being done on ecology of these fungi in temperate countries. In tropics, very little studies have been done on ecology of these fungi. So I think there is a lot of scope to study the ecology of these fungi in tropical countries. There is more information on dissemination is needed on dissemination. What we have done is very little. So there is more information required on dissemination of these fungi and the factors which influence fungal diversity. I just gave few examples of the coffee ecosystem, alley cropping systems, and things like that. And the Western Ghats experiment which we did, which affects the diversity. And so more experiments are needed to see what factors influence the diversity of these fungi, including the various agricultural practices. I listed so many agricultural practices, and we know now the intercropping is better than monocropping. So like that, and which fungicide stimulates mycorrhizal fungi. So all these can be used in practical agriculture. So there are two strategies for managing fungi. Inoculation with efficient fungi, efficient fungus. That is what most of us do, inoculate with an efficient mycorrhizal fungus. The second way of managing mycorrhizal fungi is manage the indigenous mycorrhizal fungi occurring in the native soils by judicious agricultural practices. So we have not reached that stage where we can very comfortably say this, but a lot of importance is given to this type of study nowadays. And such studies are now called as ecological engineering, where we can manipulate the native microflora to increase the crop productivity. So such studies will help to understand more clearly the ecology of these fungi. Thereby, we can manipulate them for improving the crop productivity. I am sure that the younger people here will be fascinated and take up some of these works in future. Thank you very much. Whether this might have beneficial producing growth promoting substances for host plant. Yeah.
They are asking questions.
Yeah,
Thank you, sir.
okay.
Nice presentation about AMF. Dear participants, do you having any questions? You can ask to sir, otherwise you can post on chat box.
Yeah, I can see one in the chat box. Whether mycorrhizal fungi are beneficial for producing growth promoting substance for host plants. Yes, I think yesterday I told, and today morning also I told, these fungi do produce indolactic acid, cytokinin, and gibberellin like substances, which are all plant growth promoting substances which promote plant growth. Yes.
Okay, sir, one more person, sir.
Yeah.
What is the endotrophic mycorrhiza?
Yeah, I got it. What is endotrophic mycorrhiza? See, earlier people called two types of mycorrhiza, that is ectotrophic and endotrophic mycorrhiza. The ectotrophic mycorrhiza are the ones which are all mostly basidiomystis fungi, which colonize the temperate forest trees like Betulaceae, Pinaceae, and so on. Other ones, they called as one group was endotrophic mycorrhiza. Now, this group endotrophic has been divided into many types. So, fungal, now fungi is one of the endotrophic. Archid mycorrhiza is another endotrophic. Ericoid mycorrhiza is also endotrophic. All these are jointly called as endotrophic mycorrhiza.
Another one question, sir. What is the significance in ecological importance of lichens and also mycorrhiza?
What is significance? Ecological lichens and mycorrhiza. I don't know about lichens. I've never done anything with lichens. Ecological importance, the whole lecture was on it. So you should know that.
Okay, sir. One more question. What is the deuterium?
What is?
Deuterium.
Oh, that is an isotope, which hydrogen isotope. Yeah. Sorry.
Okay, sir. One more question I am asking. Any hazardous effect on AMF in coast plants, sir?
What are the research opportunities for budding? Yeah. As a research worker on mycorrhiza, I see the opportunity is quite good. Yeah, because a lot of importance is given to the phosphorus nutrition of croplands and also reducing getting the raw material from Morocco for the manufacture of phosphatic fertilizer, thereby saving a lot of foreign exchange. So there is a lot of opportunities, yes. And moreover, our soils are deficient in phosphorus. That is a big plus point.
Okay, sir. One more question. What are research opportunities for budding research?
Yeah, that's what I answered now. Yeah.
Okay, sir. Any
Shiva,
question?
sir, you can raise your question, sir, as you told. Dr. Shiva?
Already asked, sir. Okay, anybody is having questions? No more question, I think.
Sir, I would like to ask one question. Was Bhagira, sir?
Yeah.
Sir, myself, what if most of our students from rural background,
Okay.
they are from the village background,
Okay.
they are, as per the curriculum, they have to complete their postgraduate projects, PG projects, they have to complete.
PG projects. Okay.
What are the, yes, they have to complete. Is there any idea or suggestion they can perform those things in the AMF in their own area, in their village itself?
For doing some work in their own area, in their own village.
Yes. Exactly.
Okay. Yeah, I can think over and suggest something which they can do in their own place. That should be fine. Yeah. One or two people can contact me. I can suggest something. Yeah.
Yes, sir. Sir, kindly permit me to share your email address to them, sir. They will contact you.
Okay.
Can you permit me, sir, to share your email address?
Pardon? You can
kindly
permit my
permit me
email
to,
address. No, you can pass it.
Sir, I will. Sure, sir. Thanks for the permission. Thank you, sir. Thank you. Mr. Shiva, sir, as Shivana sir joined, you can complete this session.
Yeah,
you can move on
I have
to
joined.
next session. You can thank. Yes, sir. Shiva, sir, you can proceed with vote of thanks.
Okay, sir. Okay. I take this opportunity to thank our speaker of session, Dr. T.J. Bakiraj, for his elaborate lecture. I also thank the participants for your patterns, hearing, and questions. Now I hand over the next session to Dr. R. Anathalakshmi.
Okay.
Thank you.
Thank you.
ಅಂದ್ರೆ ಅವನು ಲೆಕ್ಚರ್ ಮುಗೀತಂತ ಇವಾಗ
Good morning, sir.
ಲೆಕ್ಚರ್ ಹಾಕದ್ ನೋಡೋಣ ಹಿಂಗೆ. ಹಲೋ ಗುಡ್ ಮಾರ್ನಿಂಗ್.
Good morning. Nice.
Shivana sir, cute.
Good morning, Professor Bogiraj.
Hope Dr. Anathalakshmi joined this session. Is it Anathalakshmi is there? One second, sir, please. 1 second. Yeah, உங்களுக்கு ஒரு ஃபோன் போடுங்க.
Anathalakshmi is the
ஃபோன் போடுங்க.
moderator.
எனக்கு ஃபோன் போட்டாங்க அவங்க.
ಮಾಡ್ತೀನಿ ಓಕೆ ಬರ್ರಿ ಅವ್ರು ಇನ್ನು ಕಂಪೇರ್ ಮಾಡ್ಬೇಕಲ್ಲ ಅಂದ್ರೆ ನಾವು ಸ್ಟಾರ್ಟ್ ಮೈ ಲೆಕ್ಚರ್.
ಅಯ್ಯೋ Continue.
If she is not there, it doesn't matter. I can start off.
Oh, okay, sir. Thanks for the permission, sir. I would like to thank our convener, Professor Shivana, sir, to go ahead with his second lecture on.
Presentation.
Yes, sir.
Okay. Already ಹೋಗಿದ್ನೋತನ?
Professor is going to enlighten us with his second lecture on the sixth mass extinction, its impact on biodiversity and the human welfare.
ಇಲ್ಲಿ ಸ್ಟಾಪ್ ಶೇರ್ ಅಂತ ಐತೆ ನೋಡ್ರಿ, ಅದನ್ನ ಒತ್ತಿದ್ರೆ ಶೇರಿಂಗ್ ಆಫ್ ಆಗುತ್ತೆ.
ಇದು ಸ್ಟಾಪ್
ಈಗ ಒತ್ತಬೇಡ್ರಿ, ಲಾಸ್ಟ್ ಮುಗಿದಾಗ.
So okay. Can you hear me?
Yes, sir. We are hearing you, sir.
Hello.
Yes, sir. We are hearing you, sir.
Can you see my?
Yes, sir. Yes, sir. Perfectly, we are.
Can you see my slides?
Yes, sir. It is visible, sir. It is visible, sir. Please.
Okay.
Please, sir.
All that I can start my lecture. Now this lecture selected the topic. Yesterday I talked about one of the crises, that was global pollinator crisis. And today I have selected another major crisis, the sixth mass extinction crisis and its impact on biodiversity and human welfare. Now, being the students of biology and biochemistry, I thought, of course, the technical details you people will be very familiar, but this is something which everybody, whether he is a biologist or a non-biologist, we should come to know and see what is happening to our world and particularly to our humanity, human welfare, as well as the biodiversity and so on. Now, yesterday I commented about two of the challenges which the humanity has to face. And today, of course, I will concentrate only on conservation of.
Professor, sorry for the interference. Professor,
Yeah.
kindly go with this slide show presentation, sir.
Yeah.
Or full
The
screen.
presentation. Can you see my presentation?
Yes, sir, we are seeing, sir, but kindly show in the, click the slide show, sir. Then we can see the overall slide.
Oh,
It is partially visible.
slideshow. Where is that slideshow?
Next to this, review near to.
Remote control, mute.
No, sir. The first
Just
one is
a-
home, insert, design, transition for slideshow, sir. For,
Insert, format.
no, sir. Below to
View.
that, sir. Below to this, sir. Insert, design, transition, animation, then slideshow, sir. 1, 2, 3, 4, 5, sixth one, sir. Below to that.
Animation is not.
Yeah, this
No,
one, sir.
animation and
Slideshow.
slideshow.
Yes, sir. Yes, sir.
Okay. Can you see now? Pressed slideshow.
Yes, sir. Yes, sir. Play from
Can-
current slide. Yes,
Can
sir.
you see now?
Yes, sir. Yes, exact, sir. Play from current slide. Please press it, sir.
Okay.
Oh, that's fine. Clear,
Can
sir.
you,
Yes, sir. Perfectly right, sir. Thank you, sir. Thanks for accepting my request, sir. Thank you, sir.
now I can go. All right. You can hear me and you can see my presentations. All right.
Yes, sir. Yes,
So,
sir.
the most extinction crisis and largely its impact on conservation of our biodiversity. Now, this sixth mass extinction crisis is purely the result of human-induced environmental changes. Which is purely induced by humans. I want to elaborate a bit about what is biodiversity. Of course, those who are familiar with biodiversity, you would have seen each book if you refer or each review you refer, there will be different definitions of biodiversity. Just to define it simply, The biodiversity includes broadly all genetic variation found in all species present on this planet and their ecosystem interaction. So this is a sort of a loose definition of biodiversity. It includes plants, it includes animals, it includes microbes, and more importantly, the interaction of all these living organisms with their biotic and abiotic components. Now, how many species are there on this planet Earth? Now, of course, we have not yet documented all the living organisms present on the Earth. We have documented only half of them so far. Now, the working estimate about how many species are there right now on this planet Earth ranges from 8.7 million to 12 million species. This is, of course, the most exact estimate because there are lots of other deviations, but this is what many biologists think. This is a million of living organisms we have on this earth. But out of these, only 1.2 million species have so far been documented and named. So we are still have to discover huge number of species. Now, this biodiversity and associated eco-services provide all our essential needs. The air we breathe, the food we eat, and many of our medicines, and the major proportion of our building material and so on. In fact, practically everything we get from the biodiversity and associated eco-services. In fact, human survival and welfare depends on the survival of biodiversity or the preservation of biodiversity. Now, how did this such a vast diversity, about 10 million species on Earth, came about? So you all know, being a students of science, the vast biodiversity present on this planet is the result of evolutionary processes since the life originated on Earth about 4 billion years ago. So microorganisms originated about 4 billion years ago. And then because of the evolutionary processes, we have got such a huge number of plants, animals, microbes, and so on. Now, as new species kept on evolving on this Earth, that is, of course, as you know, it is based on natural selection, operating on heritable variations. A few species turned out to be misfits in the prevailing environment because, after all, environment keeps on changing, and on these changes, natural selection operates. I'm sorry, this. I'm sorry, I had forgotten to switch off my phone. Now, as species kept on evolving, a few species which turned out to be misfits for the prevailing environment, they became extinct. This is part of evolution. New species will keep on evolving, and because of this one, some environmental changes, a few species cannot survive and they will become extinct. However, the number of species that become extinct, which is called background extinction, because this is a routine process, was very, very, very small when compared to the number of species that kept on evolving. That is how this planet kept on increasing continuously over the geological period. So the rate of background extinction varies amongst different groups of organisms. Now, it has been calculated based on number of geological and fossil evidences and so on, it is the background extinction is 8 years, only one will be extinct out of 1 million species. So this is very, very, very small, whereas thousands of new species keep on evolving. Now, until human beings, everything was going normally. And human beings, of course, when they evolved about 200,000 years ago, they started, of course, initially they were hunters and gatherers. At that time, of course, they were hunting lots of animals and all that. But it was sustainable because the number of people were small, the population was small, and they were moving around. So the nature could sustain itself, whatever animals they killed, whatever fruits and other crops they harvested. At that time, of course, there were no crops, there were plants. So, but what is happening in recent decades is the rapid increase in the population throughout the world, combined with human greed for luxurious living, they have put heavy burden on the biodiversity and ecosystem services. So this is happening in recent decades, particularly very severely. According to the 2017 UN report, the current world population of 7.6 billion is expected to reach 9.8 billion by 2050 and 11.2 billion in 2000. And coming to our own country, in India, according to 2019 the census, we were 1.3 billion people, and this is expected to reach 1.67 billion people by 2050. So this is a huge increase in the population. So as the population increases, the resources of nature are being utilized more frequently, but more intensively. So this results in environmental changes. So the human-induced environmental changes in recent times have resulted in extinction of a large number of species. So this extinction in recent decades is much higher than the background extinctions. And they have also not only a large number of species have become extinct, but also a large number of species have been pushed into the verge of extinction. If nothing is done to save them, they also will become extinct soon. So this is what is the percentage of extinction of different groups of plants and animals. The most authentic source about how different groups of plants, animals, and microbes behave is this IUCN, International Union for Conservation of Nature and Natural Resources. This is the most authentic source. Now, the job of IUCN is they keep on evaluating different species of plants and animals on the basis of a number of criteria they have established. And based on their analysis, each of those species will be categorized into different categories. Some of them may come under extinct, that means they are no more on the earth. Some of them may be present only in ex situ, that means they may be present in botanical gardens or zoos, but they are no more present in the wild. And some may be critically endangered because soon they may become endangered, then they may be endangered, they are slightly better than that one, then vulnerable, near threatened, least concerned. These are the happier species. They are not much concerned because they survive very well without any threat. And of course, large number of species, although they try to analyze, simply they don't have enough data to analyze them to any of these things. So they put that as data deficient because more data has to be collected before they put them under any of these categories. And a number of species, and large number of species are yet to be evaluated. Now, this was in 2017, they put up a graph of these different groups of largely cumulative extinctions as the percentage of IUCN evaluated species. So out of 100,000, whatever number of species they have evaluated of mammals or birds or vertebrates and invertebrates. So this is how the extinction is, number of species that are becoming extinct, percentage rather. So this is 2,500 to 2014. You can see even particularly mammals and birds, so on, they were already, the dotted line representing background extinction as it was before this 1,500 years. Now, as we proceeded, then the rate of extinction goes beyond the background extinction. And after industrialization, they say 1850 is fixed as the period of industrialization. Now, after industrialization, you can see there is a significant increase in the number of species that have become extinct. So now the rate of extinction of species has increased hundreds and sometimes thousands of times compared to background extinction. So this is one of the problems happening, this increasing population need for these human beings for luxurious living. Now these are two of them. There are hundreds of recently extinct species. This one is the extinct species. This is one of these in one of those species, this was the last male to be alive. It's in Galapagos Islands, when there 20 of those they can live
Participant requested to be with us. Professor is facing technical issues. Kindly be with us. Sir, kindly unmute yourself, sir. Shivana, sir.
Okay, I have unmuted.
Yes, clear, sir.
Was it missing for some time or what? I don't know how it became So this became what one time, this is the most famous animal in the world because here, hello?
Hello.
Hello, can you hear?
Sir. Yes.
Yes.
Sir. Yes. Yes, okay. Sir, we can hear.
So when he died in June 2012, they didn't want to throw it or bury it and so on. They sent it to New York to restore its body by taxidermists, and they took some time, and eventually in last February or so, it is restored Lonesome George. Now it is in Darwin's Museum in Galapagos Island. I visited Galapagos Island with my family for 1 week in 2016. At that time, this Lonesome George had already died, and this has not yet come. I didn't have the opportunity to see Lonesome George, either this living or this museum specimen. Now, the other one is again this Sudan, this is the last male of northern white rhino. This became extinct in Kenya in 2018. This is one of the recently because it has been overexploited because of horns. Now coming to the plants, of course, there are so many papers.
Excuse me, sir. Sorry for disturbing you, sir.
Yes.
A slide is not available, sir. Kindly share the screen, sir. Slide is not visible.
What is happening? I don't know. Right. I don't know it is how to come out of this full screen. I'm not getting any other.
Professor, kindly permit me to share the screen, sir. I have it. I have your backup.
Okay, can you?
Oh, please, sure, sure, sir.
This,
sure,
let
sure.
Me...
Sure, sir. I will share it, sir. I will share it.
Okay, you share it. Shall I exit from this one? Okay, now this slide. Hello?
I'm started, sir. 1 second, sir. Please, sir. Within 2 minutes, I will share it, sir.
ಬೇಗ ಬರೋ ಏನೋ ಆಯ್ತು ಸ್ಲೈಡ್ಸ್ ಅವ್ರಿಗೆ ಕಾಣ್ತಾ ಇಲ್ಲ.
Dear participants, kindly wait. So we'll share the screen.
ಅವರಿಗೆ ಒಂದ್ ಸೆಟ್ ಕಳಿಸಿದ್ದೆ, ಅವ್ರೇನೋ ಅವ್ರನ್ನ ನೋಡ್ತೀವಿ ಮಾಡ್ತಾರೆ ಅಂತ ಅವ್ರೆ. ನಿಮ್ದು PPT ಇದೆ, ಪವರ್ ಪಾಯಿಂಟ್ ಇದೆ ಅಲ್ವಾ? ಇದು ನಿಮಿಷ ಕಾಣುತ್ತೆ ಕೇಳಿ.
Now it is okay, sir. Sir, we can able to see your screen as well as.
Can you see it now?
Yes, sir. Both are fine, sir. Now issues addressed, sir. Thank you, sir.
I don't know what is happening. You will see around? Okay. I'm very sorry that I don't know, something has happened and now my nephew came and he put it all right.
No problem.
Now I was talking about these two animals, that is one of the tortoise species of Galapagos Islands and this rhino, one of the rhino species in Kenya that became extinct very, very recently. The lonesome George, the tortoise, became extinct in June 2012, whereas this Sudan, one of the tortoise species in Kenya, it became extinct in as late as 2018. Now, coming to the plants, as I said, there are hundreds of such situations, so I've just taken representatives. Coming to the plants, of course, plants are not so thoroughly being worked out as far as the extinction rates are concerned, because the animals are very charismatic and people are very much interested in what is happening to animals. But plants, one thing is they are not so conspicuous about how many of them are lost and so on. Second thing, it is not so easy for plants to find out how many have become extinct. But one of the most authentic studies which has come out recently is from Royal Botanical Gardens, Kew, in collaboration with many other organizations. And this is the map they have brought out, this rate of extinction since 1900. So here you see many, the data is not available, but here they have put to show the country-wise, which is the most region where the extinction is maximum. In fact, one of the most highly threatened region is Hawaiian Islands. Here you can see 41 to 79 species of plants have become extinct since 1900. And next comes South Africa. Then the third, of course, many countries, India, part of Australia, and one of the Latin American and so on. So here, as far as plants are also, although they are not so visible and they are not studied well, the extinction rate of both plants and animals is. Now, what about the threatened species? So I talked the last two slides about those which have already become extinct. Now here, large number of species have been put under threatened category. Threatened category include all those critically endangered, threatened, and so on. Now, when compared to plant groups, animal groups, they have worked out many times more. Here, these are the number of species evaluated of cycads. They have evaluated 307 species, out of which 63% of them have come under threatened category. Now, dicards, out of this one, 36% of them have come under threatened category. And conifers, out of 607 evaluated species, 34% of them come under threatened category. Now, of course, number of species analyzed from animals like amphibians, and then sharks and rays and selected crustaceans and birds and so on are many times more. But in all of them, the extinction, the threatened rate is quite high. Now, this graph represents the cumulative extinction 2000, just quite recently, how amongst the number of species, IUCN, as I told you, they keep on evaluating, and this keeps on increasing. And this is out of those, what is the percentage of those species that have become extinct, or number of species that have become extinct. In this one, which was published in 2017, out of 96,900 species evaluated, 26,500 species turned out to be threatened. Of course, this is. And afterwards, in 2019, they put forward another version because they have analyzed large number of species, that is 112,430 species they have assessed. Out of that, 28% of them were turned out to be threatened. So the number of species which have become extinct has increased tremendously when compared to the backward extinction, and a huge, that is 28% of the species, both animals and plants in an average, they have come under the extinction category. Now, nature predicts 99.9% of endangered species and 67% of endangered species will be lost in the next 100 years. It is done. So this is a very dangerous situation. There are papers and extinction of species in all groups of organisms. Most of these reports have come from developed countries. Both the number of extinct species as well as the number of threatened species. Very few are coming from developing countries like ours. Apart from, one thing is number of species which are being pushed into threatened category. They are still surviving, but still, once we endangered and so on, we don't consider them at the level of population. Only when we say so many species are extinct, we talk of the complete extinction. No more individuals are there. But amongst the threatened one, there is a huge episode of population extinction. Those species technically may be surviving, but the number of populations and number of individuals in each population have gone down so much that they cannot take in ecosystem integrity or interaction, even if they are alive, because even if they are present, because the number of individuals is so small. So in such species, they can't sustain civilization unless the population is sufficient. So on one side, we have a large number of species that is becoming extinct. On the other side, we have a large number of species that have threatened. And also in another side, although they are still survived, the number of that means there is tremendous decline in the populations of these surviving species. Now, I don't go into the details of all the papers that are published. As I told you, hundreds of papers are being published. In one of them, this is just an abundance of tropical arthropods, that is insects. They have declined by 60-fold since 1970s. Here is one of the histogram. You can see this is the mass, because mass they determine when you take an insect net and sweep it, how many of those insects, what the weight of those insects that are collected without going into how many species are there and all those things. You can see in 96, 97, this is the number of the weight of those insects which were collected in the net, but just 2011, that is about 40 years later, 30 years later, you can see how many insects are being collected. That means the abundance of these insects has come down about 60-fold over 30 to 40 years. Now, once the insect population goes down, those animals which depend on insects as their diet, like lizards, frogs, birds, and so on, they also have gone down at different areas by 30 to 90%. Thus, there is a sort of a collapse of the forest web. In fact, this has been done in one of the protected tropical forests, this quantification. So according to the latest analysis, 40% of the world's insect species are threatened with extinction over the next few decades. Now, of course, this is only insects because they are easy to collect the data and all that. But this is true for most of the animals and wherever the plants have been investigated. Now, as I told you, there is lots of papers coming, particularly on the animal extinctions or the reduction in the abundance. Even as late as 21st January 2021, this year, there was a special issue published by Proceedings of the National Academy of Sciences, which brought together 11 reviews highlighting insect declines, principal threats, and possible action to be taken to mitigate the decline and so on. So the scientists have become so much concerned that even this prestigious journal that has brought out a special issue to highlight the decline of these insects. Now, as early as 1992, 1,700 world scientists, including the majority of living Nobel laureates, they gave the first warning to humanity. They said that they expressed concern about potential damage to planet Earth by human-induced environmental changes. They identified these are the problems they identified: ozone depletion, freshwater availability, marine life depletion, ocean dead zones, forest loss, biodiversity destruction, climate change, and continued human population growth. And these are the major causes for environmental changes. Although these people, 1,700 scientists, they highlighted this one, nothing significant happened because no remedial measures were initiated at the world level. Now, in 2017, after 25 years after the first warning, they gave second warning. World scientists, they gave second warning to humanity. This time, 15,354 scientists from 184 countries, they signed this document. They said, with the exception of stabilization of stratospheric ozone layer, as you may be knowing, earlier ozone layer breakdown over the Antarctica was one of the major ecological issues. Now this has been partly overcome. They appreciated that one. But then humanity has failed to make sufficient progress in solving these environmental changes, and alarmingly, most of them are getting worse. So this is again, they highlighted, this is what is happening. And unless the world takes some serious measures, it is going to create havoc. I explained only a fraction of this, what is happening. So the biologists agree that the world has entered sixth mass extinction crisis. So if nothing serious is done, 2/3 of the species are likely to be eliminated within a short geological time. And more importantly, they say the window of corrective action to tackle the crisis is very short, at the most 20 to 30 years. Unless you take drastic steps within the next 20 to 30 years, it will go beyond the ability to repair. So the general concept is that it is a question of now or never. If you don't do anything for the next 20 years, you will not be able to restore this crisis. Now, as you know, the Earth has undergone earlier 5 mass extinctions. Now, these 5 mass extinctions are due to natural catastrophes. Now, this gives diagrammatically these mass extinction periods. This one, as I told you, the life originated on this planet about 4 billion years back. And gradually, because of the evolutionary process, the number of species kept on increasing. The width of this indicates the number of species, so that the increase in the biodiversity. Now, the first mass extinction happened around 435 million years ago. So about 2/3 of these biodiversity species came down. Then another over millions of years, again, there was a restoration of these, and before the other mass extinction took place. But overall, over the period, gradually nature restored the biodiversity and kept on increasing. Then the worst mass extinction was this one, the third mass extinction, 2040 million years ago. In this one, 56% of the genera and 96% of the species have became extinct. Then the last extinction was about 65 million years ago, in which dinosaurs became extinct. Now, of course, with all these biodiversity increase, now we are entered the sixth mass extinction. Now, many people, the geologists and biologists, call it as Anthropocene extinction, because unlike all earlier extinction, which are due to natural catastrophe, this one is purely because of the influence of human beings. That's why they call it as this mass extinction as Anthropocene extinction. Now I some time explaining what are the drivers of sixth mass extinction crisis. So what are the causes to bring about such a degradation? One is degradation and fragmentation. So the natural habitat which used to be there, which was congenial for biodiversity expansion, was deforestation. In deforestation, that means conversion of forest land to grow plantation crops like coffee, tea, rubber, oil palm, and so on. So huge area of forest have been converted to grow these agriculture or cash crops. Then conversion of natural habitat, including forest, for agriculture, for urbanization, for industrialization, for mining, and so on. So again, the natural habitat got reduced tremendously. Third is rampant pollution and extensive use of pesticides and herbicides. This is largely after industrialization. Then, of course, the introduction of alien species which are replacing native fauna and flora. This is about the concern about habitat loss and its degradation. The other second reason, the drivers of overexploitation of biological resources beyond their ability to sustain. That's again, as I told you, the reason is because of the population increase and human greed for luxurious life, because they want to use more and more biological resources so that they can live more comfortably. This is partly because number of people keep on increasing, and then of course, all those who can afford, they want to improve their lifestyle. Then third, of course, is climate change. Now, this deforestation and extensive use of fossil fuels have led to an increase in the level of atmospheric carbon dioxide and methane. And these gases, combined with water vapor in the atmosphere, they trap radiated heat from the earth, leading to global warming. So global warming is one of the outcomes of this climate change. So these are the three major drivers which are responsible for the sixth mass extinction crisis. Now I just show you photographs about each of these. Deforestation. Now, as you know, the maximum tropical forest area we found in Amazon basin in South America. Now, this tropical forest area covers 5 million square kilometer area, huge area. With such a virgin forest, it used to be there. Now, this area, which is indicating the red zone, this is the area of deforestation since 1998. That is just the last about 40 years. How much deforestation has taken place in such virgin forest. Earlier, of course, more deforestation have taken place. Now, this is one of the recent, you can see what happens is these people who are interested to fell down trees because of the timber, they first make a road where itself large number of trees are cut. And then once they make the road, along both sides of the road, they started cutting the forest. You can see this is in the Amazon basin. This represents this one, this area. Now, this is one of the oil palm plantations. Oil palm plantation, you know, it is oil yielding plants. It is basically an African crop, and it has been introduced into Indonesia and Malaysia, and it gives oil. And now it has become their major export crop. And we import a lot of palm oil from Malaysia and Indonesia. Now, because this oil palm was not a crop of Indonesia or Malaysia, this was introduced completely. Now they introduced, what they did was the tropical forest, they completely removed and they planted these oil palm. You can see this is one of the plantations, such a huge, hundreds and thousands of acres of these forests have been removed to plant these oil palm. So this is one of the clear examples of deforestation. Now, of course, I don't have to show you the application of pesticides, herbicides, fungicides, and so on. Then, of course, atmospheric pollution. Of course, in India, a large number of cities we have a problem with atmospheric pollution. According to WHO, the particulate matter, which is less than 2.5 micrometer, the safety level is only 10 microgram per cubic meter. But here in some of our cities, particularly in winter, it reaches up to 200 microgram per cubic meter. So India, large number of cities, they suffer from high level of atmospheric pollution. Then air pollution, according to the authorities, caused 3.8 million deaths globally in just 1 year, 2016, where I could get the data. Now, water pollution, of course, I don't have to highlight this water pollution. You are all very familiar. The water pollution includes lakes, rivers, oceans, and groundwater, and so on. And of course, their impact on animals as well as human beings. I don't have to explain, you are all familiar. And the source of the contamination is untreated sewage, industrial effluents, and agriculture runoff. Now, in recent years, plastic pollution has become a dominant issue. Look at this, some of the beaches, the amount of plastic which has been pushed onto the beach. So you find plastic pollution from top of the Mount Everest to the bottom of the sea. As you know, most of the plastic is made from non-renewable resources like petroleum and natural gas, and about 6% of these natural non-renewable resources are diverted to the manufacture of plastic. And it seems about 260 million tons of plastic is produced every year, and very little of it is recycled. Most of it is disposed as waste. Plastic, of course, is not degradable, and it pollutes both terrestrial as well as aquatic environments. According to Greenpeace, around 267 different animal species suffer from entanglement and indigestion of plastic trees. Now, newspaper, the microparticles are being formed by plastic in the water we drink and so on, and its effect still they are finding out. We still don't know what is the impact of those microparticles of plastic. Now, after 2014, 15, and so on, many of the international organizations has come up. They have become more intergovernmental science policy platform on biodiversity and ecosystem services. That is IPBES. You must have heard and read it in newspaper quite often. Now, they released summary of 3-year assessment report on land degradation prepared by 100 experts from 45 countries and scrutinized by 200 external reviewers. In fact, it has been thoroughly reviewed. And this was released in March 2018. And these are some of the highlights of this report. They say that worsening worldwide land degradation is the main cause for the loss of species and ecosystem services. It underlines the well-being of 3.2 billion people. That is habitat loss and degradation. And it is a contributor to mass human migration and increased conflict. It's a major contributor to climate change and so on. There are lots of reports. And more importantly, they say the land degradation and climate change is predicted to reduce global crop yield by an average of 10% and up to 50% in some of the regions by 2050. And this result in decline of nutritional value of crop species. For example, I read a paper in rice it results in decline of proteins, iron, zinc, and vitamins. So both quality and quantity of these or crops is going to be reduced. Now coming to overexploitation of bioresources. One of them is, of course, there are different reasons for that one. One is hunting and poaching of animals. As you know, the animals are hunted for their meat, or their hide, or their medicinal or ornamental value. Sometimes they are hunted as game species, and also many times they are hunted for export market and so on. And sometimes they are hunted because they happen to be agricultural pests or predators of domestic animals. And many of them, like birds and some of the reptiles and so on, they are collected for pet trade. This overexploitation is being done not only for local use, but also for export market, because they get huge money by selling these ones. According to an UN estimate, about 30% of the global species threats are due to international trade. Now, this morning, I saw one paper published in Nature, Ecology and Evolution. This was published in the issue of only day for yesterday, March 15, 2021, in a very prestigious journal, Nature, Ecology and Evolution. And they analyzed, in this paper, they analyzed 31 papers related to trade-driven decline in 452 species of mammals, 36 species of birds, and 18 species of reptiles, and found that 76% decline of these animals was due to national trade, and 66% decline was due to international trade. And this decline of animals happened even in the protected area. It is not just in the open forest. Even protected area, these things happen. The global wildlife trafficking, it seems it generates US $5 billion to $20 billion a year. That is the huge amount involved in this animal trade. Now I'll just give you two examples. No, I have to hurry up. One is Himalayan musk deer. It occurs in high ranges of Himalayas. Male one, it produces musk spot in the abdomen. This gives persistent aroma. There is a great demand in cosmetic industry of this musk spot. And I understand according to IUCN, each kilo of this one fetches $45,000 US. So there is great demand. So the animals are being poached extensively in Kedarnath Wildlife Sanctuary. So now it is very difficult to find this male musk deer. From outside, you can't distinguish whether it's a male or a female. So they kill. If it is female, they throw. If it is male, of course, they harvest this one. That is another problem. Now, the donkeys were quite comfortable. All these years, nobody was bothered. They used to be used to carry luggage and all that. But now, recently, China has started using the hide of these donkeys for preparing some important medicine. So now, in most of the African countries, donkeys are, and they, for the past 6 years, Chinese traders have been buying the hide of millions of butchered donkeys from countries, particularly from Kenya and other African countries, because they use that hide for the manufacture of medicinal medicines, which is very popular in China. Now, donkeys are also threatened now because of the exploitation of their hide. Now, coming to plant, of course, our major problem is legal and illegal timber extraction, because collection of, and also apart from timber extraction, collection of non-wood forest products like gums, resins, poops, and sources of medicine and so on. So all these things we collect in an unsustainable way from the natural forests, and many of these medicines particularly are being exported, although they are collected from natural forests, not from the cultivated plants, from natural forests. So these species are being threatened and they are getting critically endangered, but we are extracting them for export. And another thing is, for example, timber. The tectona, you know how important it is as a timber. In Ecuador, a small country in South America on the Pacific coast, teak is not a native to this Ecuador. Now, because the India can import lots of teak, and they are destroying the forest and agriculture land, they have started growing teak purely for export purpose to India. So like that one, in fact, India and China are the major markets for illegal timber trade from Malaysia. So this illegal trade flourishes. Then, of course, climate change. Increase in atmospheric CO2 level result in increasing mean global temperature. And this results in the disappearance of most remaining glacier because of the increase in temperature and so on. Rise in sea level, submergence of small islands and most of the coastal areas. And of course, when this happens, biodiversity of such islands, when once the island submerges, then of course you can't conserve it because which will happen in a very rapid pace, even in the coastal area. Now, this is just a few islands are there in the north of Australia, between and one of them is Mosik. This is the Mosik Island. In fact, rise in sea level, every year, 1 to 2 feet from all sides, water will enter and people keep moving to the center. And now, in another 2 or 3 feet, water goes up, the island will not be there. Like this, these aborigines, they have gone to Europe. Australia is not this global warming and so on. Examples. Now, this increasing atmospheric CO2 the atmosphere, it also dissolves in seawater and also content. More importantly, it makes this pH like danger of increasing temperature, and this acidity of seawater is very dangerous for the animals, marine animals. One of the things, of course, coral reefs are very important, both from tourist point of view and so on. Corals, you know, they are the organisms between algae and one of the animals. And when once temperature goes up to Fahrenheit, then the algae will get killed, and then the coral will bleach like this. Then it will die over the years, it breaks down. Now, Great Barrier Reef, you know, in Australia, it fetches large number of tourists, and this has about 30% of that one, or 1/3 of Great Barrier Reef of Australia already bleached. Unless something is done, probably it will continue to bleach. Now, this is just a healthy part of the coral reef. And this you can see part of the bleached coral reef in Great Barrier Reef, which is become completely dead. Now this Great Barrier Reef provides 70,000 jobs and billions of dollars to Australia by tourism industry. And when once it becomes like this, then all this is. This is the part of Australia, this is Papua New Guinea, and this is the Great Barrier Reef. Now, this is the UNESCO World Heritage Coral Sites. They are shown here. And it shows all the red ones are severely breached, more than 30%. Yellow ones are moderately breached. Only those small, a few of these blue ones, they are not yet bleached. Now, the other thing, apart from corals being dying, they provide suitable habitat for other species, including sharks, turtles, whales, and series of fishes, and so on. So when the coral reef or coral dies, the whole ecosystem will collapse. Now the intergovernmental climate change, this is another see, they released the fifth assessment report in 2014. They projected the increase in mean global temperature. If it goes as of now, it reaches 3.7 to 4.8 degrees Celsius. It increases when compared to pre-industrial period by the end of this century. Pre-industrial period is 1850. If there are no new policies to bring down this global warming. Then in 2015, all the governments of the world, they met in Paris to discuss the climate change and what should be done and so on. So in that meeting, they formally agreed to limit temperature increase to 1.5 degrees Celsius above pre-industrial level by the end of this century. As it is, as I said, it is 3.7 to 4.8 calculated, but they wanted to control it to 1.5 degrees. But soon they realized it is almost impossible, but they said, even if it goes beyond 1.5, but we should make sure that it will not go beyond 2 degrees. That is the general concept of the world about the Paris Agreement. As I said, the present trend indicates the increase is going to be more than 3 degrees Celsius. And of course, this report, apart from this one, it continues to highlight the loss of biodiversity and other catastrophic related climate change and so on. Now, UN report on climate change, they released the latest report in October 2018, this in South Korea. They gave a final warning to the world. So this report was prepared by more than 90 authors from 40 countries, and it was examined by 6,000 scientific, they analyzed 6,000 scientific publications, and the report underwent reviewers, 1,113 reviewers. Now, this report, which was the latest, 2018 October, it was released by the UN agency. It warns that overshooting even 1.5 degrees Celsius will be disastrous. It will have a devastating effects on ecosystem, community, and economies, and so on. By 2040, the report predicts there could be global food shortage, the inundation of coastal cities, and a refugee crisis unlike the world has ever seen. Even 1.5 degrees Celsius warming would rise sea level by 26 to 77 centimeters by the end of the century, and 2 degrees Celsius, because now, as I told you, most of the countries, they were ready to go up to 2 degrees, but not beyond 2 degrees. But they say even 2 degrees Celsius itself would add another 10 centimeters, which would affect another 10 million people living in the coastal region. Coral reefs are projected to decline by 70 to 90%, even at 1.5 degrees Celsius rise. At 2 degrees, 99% of the reefs would be ravaged. So as we have been seeing for the last many years, storms, floods, droughts, and forest fires would increase both in intensity and frequency. So this is the warning by the UN. And they say, this report says, there is no time. It has already reached, the temperature has already increased by 1 degree Celsius now. According to Paris, it should be 1.5, but already 1 degree has already reached now. They are currently heading for about 3 to 4 degrees warming by 2100. Now, if they want to bring it, cut it until 1.5 degrees Celsius, the coal share to no more than 2% by 2050. So renewable power then to keep 1.5 degree target, net anthropogenic CO2 emission must come down by 45% from 2010 level, at whatever was by 45% has to come in by 2000 and reach net zero. That means it has to be equal to 2010 levels by 2050, which appears to be almost impossible. Now, the report, again, UN report highlights the world cannot afford a 2 degree Celsius. So this decade, they say that is 2020, now we have started. The next 10 years, the humanity's cost were devastating impact. So if at all we have to do anything seriously, it has done next 10 years. Otherwise, it will go beyond our ability to do anything. Now, more than 11,000 scientists around the world clearly and unequivocally, I told you earlier also two that already given international scientists. Now it is 11,000 scientists, clear warning, unequal warning. They declared that the planet Earth is climate emergency. In fact, they have given series of problems humanity have highlighted only some of them so that you can follow it. From 1980 to the present, look at how the population is increasing, how the CO2 emission is increasing, how the atmospheric temperature is increasing, and how the Arctic ice is melting, and Greenland ice is melting very, very fast. Antarctic ice is melting very fast. And these glaciers are melting very fast. Himalayan glaciers, there have been many papers to say Himalayan glaciers are melting. And we recently, we had that Uttarakhand problem where one of the glaciers melted and fell down, and all that tragedy where large number of human beings died and there was a lot of damage to the property and so on. So these scientists, they say it is more severe than anticipated, because earlier there was some sort of a concept telling this much will happen and this much will happen. But now they say the severity is much more than what we had anticipated earlier. So according to them, nothing else will work unless, if we want to secure a sustainable future, we must change how we live. Otherwise, we can't do this. This can't happen. Now, coming to the conclusions, the impacts of human-induced environmental changes are all around us. We know every day, even in our own country, how the droughts and floods are occurring in different places, how in our area, the monsoon is reducing. Now, it is important for every one of us to do our best to reduce human-induced environmental degradation, to preserve, to protect, and conserve our biological diversity. This is imperative to protect ourselves, our children, and our planet. So we need more and more useless people, not useless people who can't do anything, but useless people means those who use less resources of Earth, so that we have to start remedying the 6 mass extinction crisis or the impact of human-induced environmental changes. Now we should remember the Mahatma Gandhi's statement. Earth provides enough to satisfy every man's needs, but not every man's greed. This is a famous statement by Mahatma Gandhi, often quoted in different occasions. Now, the more important, particularly the younger generation, should realize the present generation is the first generation to face severe impact of climate change. We have started feeling the climate change. Earlier, probably people were not feeling because it was not that apparent. Now it is also the last generation capable of taking effective measures to reverse its impact. Now, the bottom line is time is running out to save the earth. So the world has to think of this seriously and see what best we can do. Now, as you realize, for the last couple of years, how many floods we get, how many tornadoes you get, how many cyclones we get, what is the intensity of forest fires, for example, in California, as well as in Australia and so on. So you take of any disaster, they are increasing both in intensity as well as frequency. So unless we take drastic steps, marginal steps will not do. Unless the world takes drastic steps to curtail this one or to mitigate this one, then it will be beyond our reach. Now, of course, we know how such a vast biodiversity arose on this planet Earth, because it arose because of the Darwin's theory that origin of species by means of natural selection. Whereas now we know how these species are being destroyed by anthropogenic activities, the mass extinction of species by means of environmental destruction. Of course, now it's a hot topic wherever you go. There are many books which have been written earlier also about the sixth mass extinction crisis. And recently, I have written an article on sixth mass extinction crisis and its impact on biodiversity and human welfare in the later issues of Resonance. That means last issue of Resonance. So as I told you yesterday, this Resonance is available to anybody. You don't have to subscribe it. You can download any article in Resonance of any issue. So all the issues are available in the internet. So if you are interested, anybody, you can go to that one. You will get a bit more information on that one. So with this one, I will thank you for your patience and most welcome to ask questions and any clarifications and so on. Thank you very much.
Thank you, sir.
Hello. Yeah.
Dear participants, those who are having questions or doubts, you can post your questions in the chat box. So we are having such an eminent personality here to clarify our doubts. So you can post the questions. So already some of the questions are there, sir. Shall I read out, sir?
Okay.
Sir?
Yeah, I don't know. I didn't find the chart. Yeah, please,
Okay, sir,
one by one, you read out.
Okay, sir. I will read out. So the first question, how human actions contribute to the sixth extinction? How human actions contribute to the sixth extinction?
The whole lecture is based on what are the things which human beings are doing to bring about the sixth mass extinction crisis. One is the destruction of the habitat by deforestation and the destroying natural habitat, then overexploitation of the species, then of course the climate change. Even climate change is induced because of the human beings, because you are using too much of fossil fuels, you are removing the forest which used to be absorb carbon dioxide and produce more and more of oxygen, and all these things is increasing the atmospheric CO2, and that is responsible for increasing the temperature of the atmosphere, and that indirectly results in the ocean and increase in the acidity of the ocean, which are destructive for the marine life. And also look at the pollution, what is happening. The pollution, whether it is atmospheric pollution, whether it is water pollution, whether it is soil pollution, whether it is plastic pollution, all these are destroying the habit, and they are harmful not only to human beings, but also to all the animals. So like that one, in fact, my whole lecture is based on how we are spoiling the environment so that the sixth mass extinction crisis has come around. All these, what I explained, is because of the human beings. It was not because of the, it has not been because of any of the animals.
Thank you, sir. So Ashik Ahmed, he has requested for some endangered species in recent years, sir.
I told you I have given you two examples, and again there are a large number of birds. If you go to internet, of course, I can't offhand tell you here. In fact, in my resonance articles, I have added some more animals in that one, but I have cut them. I have taken only two just to highlight the recent extinctions. If you go to internet, because now you people, youngsters, are lucky because you can get any information you want by just your fingertip. You just go to Google and type whatever you want to get the information. Yes, there are a large number of animals, particularly we have data for animals. Plants, it is not so easy to get clear-cut the data, but animals, you can go to internet and get the whole list of animals which have become extinct in recent years. Yeah, any more?
Yes. Yes, sir. Yes. So next question from Dr. Jayprakash. What is the impact of technology on diversity loss, especially to the bird population due to mobile phone towers?
Okay, of course, the bird population is going down due to so many other reasons. Global warming, and their diet is being affected because number of insects are going down, and their density is going down. They don't get enough diet, and also because of the warming, they may not be able to sustain and reproduce and so on. Then apart from that one, now what is the question again? I just went off track.
Yes, I will repeat, sir.
What is the question?
I will repeat, sir. What is the impact of technology on biodiversity loss, especially to the bird population?
Okay.
Due to mobile phone.
About mobile tower you were telling.
Yes, yes.
Mobile tower, of course, many newspaper reports and magazines and so on, they are affecting bird population. But actually, we don't have too much of critical data with enough controls and so on. Because quite a bit of that one is more of a perception rather than scientific demonstration as far as mobile towers are concerned. Because if I go to, in fact, I did search in the internet, we don't have a very clear-cut demonstration in terms of scientific papers which has been published in a reputed refereed journal. Most of them are a sort of a general write-ups without any clear-cut evidences. So I'm not telling they don't have, but we need to study more clearly with effective controls and so on. So right now, it is very difficult to say whether it has any significant impact, particularly in terms of radiation. Of course, sometimes quite likely they may get affected because they may hit the tower and some of them may die and so on, but that is not the major criteria. So as far as that radiation of the mobile towers are concerned, at least I have not come across any paper which has been published in a reputed, scientifically reviewed journal demonstrating it has a serious consequences on bird population. Thank you.
Thank you, sir. So next question from Niveda. So will human go extinct? Will human go extinct?
Yeah, it depends on what type of action we take in the coming decades. In fact, if the same trend continues as it has until now, human beings also may become extinct in another couple of 100 years. It is not immediately, but if you take suitable action, very drastic action, and as the scientists say that unless we change the way we live, then probably we may be able to restore. But otherwise, in fact, somehow whenever I think seriously, I feel that probably human race has thrived a lot, but it has already reached a peak. And the next stage is their extinction. So I don't know. It depends on how the new generation. It depends on how the new generation behaves. Not only in one country, but the whole world has to behave as a unit to overcome the problems of sixth mass extinction crisis. Because unless we preserve our biodiversity, we can't sustain ourselves. Okay,
One second, sir.
Yeah.
So next question, sir. So why government should not be strictly focused on in situ and ex situ conservation scheme?
Professor, kindly unmute yourself. Sorry for the inconvenience. Kindly unmute yourself.
Okay, now can you hear me?
Yes,
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 sir.
we are hearing, sir.
Okay. Now, one of the major conservation program is in situ conservation. That is to come up with large areas as protected areas, like national parks and reserves and so on. That is happening throughout the world. Even in India, we have large, huge areas of protected areas, whether it is national parks or national reserves and so on. But the implementation is the problem. Because implementation, there are lots of problems. Illegal felling of even the protected area is taking place, not only in our country, even in the Western countries. As I said, even in the protected area, things are happening, the hunting and many other activities which are not supposed to happen. So that is the main in situ program. And also, in situ program, just because you make hundreds of acres protected, that doesn't mean unless you take care that they are really protected, they are not protected. So also what happens is, first of all, we have to understand, as long as plants are concerned, what is their limitation, why they can't sustain. Why they can't sustain is because they can't recruit new individuals to the population. So after all, the sustenance is manipulating or managing the ratio between number of deaths and number of births. So in the population, some individuals are bound to die every year. And in a sustainable population, new individuals come up to replace the individuals which die. But when once number of deaths increases over the number of births, gradually the population will go down, and eventually, if nothing happens, it will become extinct. So one population may become extinct now, in another 5, 10 years, another population like that, one whole species may become extinct. So why should they become extinct? Although climate change and all these today I explained are the proximate causes for extinction, the final causes are that these things will affect the ability to recruit new individuals as far as flowering plants are concerned or any other plant is concerned. Now, coming to the flowering plants, These human-induced environmental changes create lots of problems in terms of if there is a new recruitment of an individual, it has to start with pollination. Yesterday, I told you. If there is a problem of pollination, then there is no recruitment. And sometimes if the temperature becomes high because of the climate, global warming, then pollen fertility is affected. Then even if pollination takes place, pollen grains will not be able to germinate. There is no fruit set and seed set. If there is no seed set, there is no recruitment. And sometimes even if fruit set is there, if all the seeds and fruits fall down on the tree, they cannot survive. The new plant cannot, even if it comes up, it cannot thrive because there are lots of shade, there is a lot of competition and so on. That's why some of these seeds have to get dispersed, either through animals or through other agencies. They have to get dispersed and there they have to germinate, and then the germinated seedlings have to get established and new young adult has to come up. So the human-induced environmental changes will affect the ability of these plants to recruit new individuals by bringing some problems of different stages of its reproduction. And that is the main criteria, whether it is for plants or whether it is for animals. And that is how it is affecting human-induced environmental changes for the biodiversity of these, whether it is the plant or animal, whatever the case may be. Okay.
Thank you, sir. So next question is from Pavya. What happens if the asteroids hits the Earth?
Asteroid hit the Earth, that is one of the theories why dinosaurs became extinct. Because according to theory, again, you can go to internet, you get all the details. When once the asteroid hit the Earth and if it is reasonably large, then it creates complete darkness because it raises lots of dust and so on. And if it continues, then of course, the plants will not be there, its food will not be there and all that. So there are different theories. Of course, nobody has demonstrated this asteroid is responsible for the extinction of this dinosaur or any other organism, but these are theories. So one, you can, if you want more details, you go to internet and you get all the information. Yeah, earlier extinction, some at least people say that it is due to asteroids or any other. For example, even our human civilizations, take for example Harappa culture, which we are all familiar. Now there are many papers are coming up to say Harappan culture vanished, which was so thriving at one stage because of the continuous drought, it seems. They are getting evidences to say that because of the continuous droughts, they were not having agriculture because crops were not growing. And at that time, of course, there was no mobility. They could not move far beyond where there was rains and water and so on. So eventually, the whole civilization collapsed. Like that one, there are many examples. In Mexico, in Latin America, they have come up with lots of evidences to show it is because of the human-induced environmental factor. There are many areas, islands in Pacific. There was one island, I forgot the name. Anyway, what happened was, it was a thriving island with lots of forests and all that. Then gradually they removed forest. Over a century or a couple of centuries, all the forest trees were removed. Then it became, the rains became less and less. And then there was a drought. And eventually, lots of people started dying. And all those, some people who were left out, they wanted to use the boats and migrate to the neighboring islands. Of course, neighboring island means a couple of hundreds of kilometers and so on. But they didn't have boats. And they could not build boats because there were no trees. They had removed all the trees. Then eventually all of them died like that one. There are very number of examples. In fact, there are many books which have been written on this one what happens to those old civilizations which collapsed. And if you are interested, you should go to that one and try to read. They are very interesting. Okay.
Okay, sir. So the last question from Dr. Parameshwari. This current pandemic issue might be a human-induced changes, and in future, can we expect like this, sir?
In future, the thing is, as long as human being is there, he won't allow any other species to become dominant. By chance, human beings become extinct if they don't take care of the situation. Then some other species will become dominant, and we don't know what is that species going to be, whether it is human beings themselves completely modified or something, or some other species will come up and so on. So probably it may undergo another changes. It will take maybe a couple of years before that becomes dominant, and again it starts having problems with the environment and so on. Then again, so the cycle may start. In the sense, this is all imaginary. We don't know how things go. But as far as the present scenario is concerned, it is not surprising that in another 200 to 300 years, human beings may become extinct unless they take drastic steps to restore or to bring out remedial measures, all these human-induced environmental problems. Okay?
Thank you, sir. Okay. So I would like to thank Dr. K.R. Shivana, the convener and the backbone of this virtual lecture workshop, on behalf of PG and Research Department of Biochemistry, for his outstanding delivery. It's our great honor to have such an eminent personality with huge knowledge and experience both in teaching and research among us. Before winding up the session, let me to share few words about Sir. So actually, Sir was a retired professor of University of Delhi with 35 years of teaching experience and more than 55 years of research experience. He has guided more than 23 PhD students and 9 MPhil students, and he has published more than 200 original research papers and review articles. So this is what I want to highlight for the new entries, new joiners of the session. So a new plant species, Combretum shivanae, was named after him in honor of his outstanding contribution in the field of plant reproductive biology. So we had such an eminent person among us, and we are grateful and also honored to listen to his lecture. So learning is synthesizing seemingly divergent ideas and data. So once again, I thank you, sir, for being with us and delivering such a wonderful lecture with us.
So thank you once again for all of you and all the participants, and I wish you all the best. So may God bless you. Thank you.
Sir, is it possible, can you join with us in valedictory, sir? Or
Valedictory at what time? Is it? Yeah, it should be possible. What time?
4:30 to 5, sir. 4:30 to 5, sir. 4:30.
Okay. Yeah, should be possible. Yeah.
Okay. Okay, sir. Thank you.
Yeah,
Kindly
thank
make
you.
a time of it and join with us, sir. Thank you, sir.
Yeah.
The same link, sir.
4:30 I will.
Thanks, sir.
Yeah.
Thanks for
Thank
you.
you.
Okay, sir. What
All
Ananta
right.
Lakshmi, ma'am?
Bye.
So education is the ability to listen to almost anything without losing our temper or our self-confidence. Thank you, dear participants, for your patient listening and active participation. So let us join the next session after the lunch sharply at 1:30. Thank you, one and all.
Thank you, sir. The participant can leave this session. You can join as ma'am instructed. Thank you.